it's an engineering sequence and stimuli dependent. Oh wow, this is a good one.
Yeah, long
So
time.
I'm going to not read this. It's about yeah. So it's presented by Roger. So,
Yes, sir.
yeah, go ahead.
Sorry about the long title, but I will try to explain my best. My name is Rajat Chauhan, and I'm a postdoc in Dr. Martin now. And here we go. So this is my outline. I have divided in the way so you can understand this easily. My introduction is doxorubicin, which is the key premises of this talk. Then anti-nuclear aptamer, it's a DNA, a gold nanoparticle, and what design we have done to make sure this innovation works. Some materials and methods, sequence-dependent size and stability analysis, a sequence and stimuli-dependent doxorubicin release, and some future work. Okay, so what is doxorubicin? Actually, this was Dr. Martin O'Toole's idea. The docs can intercalate with DNA or RNAs, so use it for something to build on your system. So if you look, I don't know where to stand exactly, but I'll try to explain it. So there is a representative RNA intercalation you can see over here with doxorubicin intercalating in the RNA 16. And that's what we want to use. This ability of doxorubicin we want to use. What it does, it can make sure you will have a intracellular and extracellular control release in your cellular matrix. And what outcomes we are expecting, multi-drug resistance, of course, it can control, cumulative toxicity, it can control, some adverse effects, we are just claiming it will do. But we are right now at just a prototype stage. This is doxorubicin, it's intercalating. So that's what we are going to use. This is aptamer we are going to use for, we have some research done, and if it's successful in cell studies, we are lucky. But this is the very core part of this talk. This is an anti-cancer aptamer which intercalates with dox, or dox intercalates into aptamer, sorry. It's a quadruplex aptamer. It have a G quadruplex structure. G is a guanine. It's a 26 base pair long sequence that we use, and it creates a compact aptamer, and it have a anti-cancer properties. By anti-cancer properties, I mean it bind something called cytoplasmic nucleolin. There are so I'm a chemist by training. I don't have much idea about cell studies, but this is what I learned over a period of 3-4 years. So there are 3 kinds of nucleolin in the cells, in the cancer cells: nuclear, plasma, and cytoplasmic. So what happens is this aptamer goes through the plasma membrane and goes and binds to cytoplasmic nucleolin. And this cytoplasmic nucleolin is a bad guy. It just helps the oncogenic RNA to increase. So once this aptamer binds to it, it starts the anti-proliferation activity. So what Dr. Bates' group have found out, it's a selective accumulation in cancer cells, anti-proliferation activity, low cytotoxicity, and no inhibition of nuclear nucleolin. So that doesn't happen in normal cells. No plasma membrane nucleolin is there, no cytoplasmic nucleolin is there, no nuclear, and it does not harm the nucleolin. So what I did is, when I joined Dr. Martin's lab, is I made some gold nanoparticle. Previously, people were making 5 nanometer or 6 nanometer size particles. I just made 4 nanometer, and I make it like 99.99% monodisperse, highly stable. So it's a drug delivery vehicle. And what you're seeing over here is a very simple protocol. 15 minutes, it's done. Add water, 95 ml. Add citrate, add sodium borohydride, and this is ready. Like, it takes 3-4 months of training, but it can come down to as simple as that. And once it's ready, we just freshly prepare it and load it with aptamer. By loading, I will say it's the maximum loading we do with a thiolated aptamer. It have a thiol moiety, it binds to the gold covalently. And we have DNA and its control sequence. By control, I mean that was a guanine-rich cancer-targeting aptamer, and its control is a cysteine. We replace guanine with cysteine. And we load it around 18 to 20 oligonucleotides aptamers per gold nanoparticle. This is what we have done. For last 3 years, we have built it. This is a schematic what we have developed. So if you take this gold with the aptamer around it and a control sequence around it, and you intercalate the chemotherapeutic drug across it, this purplish thing, I'm saying intercalation, non-covalent intercalation happening to the, whether it is AS1411 or whether it's CRO. Okay? So what happens after that? It's easy, just incubate it overnight and it's ready, right? So what happened after that is just a quick boring slides about materials. This is exactly what we did. Everything is here. I use these reagents to get the materials and have the protocol ready and use these aptamers. One is a DNA binding cancer targeting aptamer AS1411. You can look at its sequence, mostly GGG guanines, and its control sequence, C's and T's. And then we have doxorubicin, which was in our refrigerator, and we intercalate it. And we use 5 or 6 methods to characterize it. First method, to find oligo loading on the particle, UV vis. We use a DTT cleavage method by cleaving the aptamer from the gold surface. And we measure it with the UV, and lots of other things we do with UV, like surface plasmon resonances and like measuring what the particle is, it's stable or not. There is a whole another field of knowledge around it. Dynamic light scattering, measure the size. Zeta potential, measure stability. TEM measure size also, but in a more accurate manner, like more resolution, better resolution. And fluorescence studies were performed on the modulus fluorimeter in SRB in Belknap campus. And we use the emission that is correlating with the doxorubicin. So what we find out is a size and stability based thing. Over last 3 years, we are repeatedly seeing this almost every second day. What we are seeing is a gold nanoparticle have citrate coated, which I make within 15 minutes, I was telling you. If I coat it with the quadruplex aptamer, it have a hydrodynamic size of 17 nanometer, like 13, minimum 14 to 17 to 19. But once it goes to CRO, it becomes at least 20 nanometer plus. Like even if the instrument is not behaving well, it's plus 20 nanometer size. So once you coat AS1411 and CRO 1:1 ratio to this particle, then you have a size at least bigger than the AS1411. So this is what we find, that the size is dependent on the sequence, just because it's a compact thing, right? And another thing is stability. The stability of this is measured with the zeta potential. Stability means if you put this solution overnight. Sometimes I come to the lab and I see, when I started this project, it was like a 1 liter bottle. Burgundy red, looks like wine, and next day I come, it's a colorless solution with a solid chunk of material sitting at the bottom, because it's a PBS solution and it's not working. So we slowly graduate that, make the optimized conditions, and now it's working. And we find out over a period of time that, oh, it's a sequence-based stability. Like CRO is introducing a better stability. You can look at the zeta potential, 52, like never less than 40 millivolt of zeta potential. Pretty strong electrostatic repulsions between particles. So that's what we find, size and stability, sequence-based. Here is a TEM data that I'm showing. Size is, sorry, it's not micrometer, it's nanometer. I'm sorry. Another thing is, it's 4 nanometer it's showing, because the core shell, it's just showing you the core shell but not showing you the DNA, because we haven't stained the DNA with uranyl acetate. We are doing this experiment and we will do it. Okay. One more slide. That is what we find out what happened with doxorubicin. Doxorubicin is non-covalently bind intercalating, very small, very sensitive material. We need to stimulate it, right? But stimulation means shake it, filter, heat, gamma radiate, or we can do the DTT cleavage, which is the harsh method, right? And what we find out that if we do 1:1 ratio of this DNA and its control sequence, then your release is almost completely enormous in comparison to if you just coat it with the regular ones. Like if I coat it with anti-cancer aptamer or the control ones, almost nothing happens. But if you take 1-to-1 ratio, something synergistically happen and it just release the doxorubicin in a very good manner. That's the innovation. That's what we find out playing around with this concept for the last 2 years. And in conclusion, size and stability are dependent on the compact structure of the, size is depending on the compact structure, stability is depending on this control sequence, like jumping too high. Dox is releasing if you add 1-to-1 ratio, and mode of Dux binding is through intercalation. And our future work is just changing the ratio first, like changing AS1411 to CRO 1 to A1, 1 to 0.8, 0.6, 0.4, 0.2. And then if something works, then try in the cell studies, and if it works, try in the animal study. We don't know where that path is going, but that's what future is, right? So, thank you. I want to acknowledge Dr. Martin for today.
Good on time. Question. Plenty of time for questions. Let me ask one. You have a picture showing, go back to your picture. The size doesn't look like that uniform yet, that picture. You know, particularly the lower one. Almost there's one like 3-5 times bigger than the small one.
So mostly the size.
No, picture. I'm talking about, yeah, TM picture. Is this TM or SM?
TM.
Yeah, see that? Lower one, you see like the biggest one is almost the bottom one, yeah, there's a big particle there, and then you have a smaller particle. So what you could do to improve the uniformity?
So the way we analyze uniformity, first is the monodispersity of the material. So it's like its PDI value, polydispersity index. It's pretty small, and we got this value in the solution. Like if it's less than 0.7, the PDI value, then we assume that it's monodispersed. It's 90% or more is one particular sphere. But as you ask about the TEM, what I'm thinking is we have like roughly 200 images collected for per particle, like gold AS1411, and I just put the representative image. Like there are plenty of images you can see with the sphere showing similar. I just didn't put that. Like this is like little more honesty, like just showing it. Right?
Any other question? We may have time for another question. Okay, good. Thank you. Let's move on. Next one is also from Louisville. Predictive biomarkers for
neoadjuvant.
Neoadjuvant, yeah, that's the one. Chemotherapy response. Okay, yeah, go ahead.
I am Sham Abdel Tawab from University of Louisville Bioengineering Department. My talk is a survey about muscle invasive bladder cancer patients and their eligibility for pre-operative chemotherapy. Here are some statistics for bladder cancer in the United States. Bladder cancer is the 5th most common cancer. In 2017, more than 79,000 patients diagnosed with bladder cancer and almost 17,000 dead. And there are more than 700,000 survivors with bladder cancer. One of the challenges of bladder cancer is that it has a high recurrence rate. Approximately 50 to 80% recurrence rate, and it's more common among males more than females. In order to diagnose bladder cancer, the urologist make a cystoscopy in order to examine the bladder cancer, and he might take tissue samples for analysis. Also, he might do a radiology tests such as CT scan to see the bladder and to determine if there are cancer has spread to other locations. The diagnosis will help him to determine the stage and the grade of the bladder cancer and determine the best therapy for the patient. The diagnosis can be divided into two categories: non-muscle invasive and muscle invasive. Non-muscle invasive found in 75% of the patients, and the muscle invasive category found in 25%. By the time the non-muscle invasive advances to muscle invasive bladder cancer. And my survey is focused on the treatment of muscle invasive bladder cancer. The typical therapy of muscle invasive is radical cystectomy followed by reconstructive surgery. However, even after the radical cystectomy, half of the patient die. And in order to increase the survival rate, neoadjuvant chemotherapy or preoperative chemotherapy has been recommended to control the cancer. And the literature has shown that neoadjuvant chemotherapy increases the survival rate in 5 years. And now the European Association of Urology recommend neoadjuvant chemotherapy, especially cisplatin compound. However, there are challenges in using the preoperative chemotherapy. It has been shown that only 40% has complete response, because there are chemo-resistant patients who don't have any benefit from having the chemotherapy. And also it has been shown that having only the radical cystectomy is better for you than having the neoadjuvant chemotherapy. And my survey is surveys the current approaches to answer this question. What are the predictive biomarkers that will enable me to identify the patients who will not get any benefit from the preoperative chemotherapy? Usually, the studies extract bladder cancer cells from the bladder and extract their genetic information for differential analysis between the chemosensitive and chemoresistant in order to find the biomarkers for chemotherapy response. The first biomarker that was explored is the DNA repair genes. Cisplatin is one of the chemotherapy drugs that works on making damage in the DNA by causing cross link on the DNA, which deteriorate the DNA function. This damage is repaired by the DNA repair genes. But what if there is a problem in the DNA repair genes? This might enhance the effect of chemotherapy. The studies investigated this point, and it has been shown that one of the genes, ERCC1, DNA repair gene, it was found when there is low expression of this gene. This will help the patient to be chemo-sensitive. And another gene is ERCC2. Somatic mutations in these genes were found to be significantly enriched in cisplatin-sensitive muscle-invasive bladder cancer patients. Another study investigated the mutations in 3 genes, and it has been shown that mutations in these 3 genes found in about 87% of the patients who respond to cisplatin-based combination chemotherapy, and it were absent in the chemo-resistant patients. Non-coding biomarkers, the genes who, that do not produce proteins, that were also explored, and here are some examples of non-coding biomarkers such as MIR200B that had a significantly low expression in muscle-invasive resistant cells, and introducing exogenous MIR200B to resistant cells makes them more sensitive to the chemotherapy. Another example is MIR223' had a significantly higher expression in chemo-resistant bladder cancer cells. Also, the long non-coding RNAs was also explored, and here is some example of non-coding biomarker that induce the chemo resistivity and significantly reduce the overall survival. Also, the tumor suppressor genes, which prevent the formation of cancer by regulating cell cycle and initiating apoptosis at a time, was also explored. And here are some examples: B53 and GRIM19 was found to be significantly downregulated in bladder cancer patients who showed tumor recurrence due to non-response to cisplatin first-line therapy. Also, there are many examples in the literature, such as BCL2G. Epigenetic regulation has also a contribution to chemotherapy response. A study has shown that the methylation of a gene called HOXA9 had significantly high levels in cisplatin-resistant cells compared to sensitive cells. This study also tried to make the bladder cancer more sensitive and to sensitize the resistant cells. They added inhibitor to the methylation, and this resulted in increased cisplatin sensitivity in resistant cells. Also, decreased the expression of two genes, ESS1 and SET1, due to hypermethylation, were found in cisplatin-resistant muscle-invasive bladder cancer. And decreasing the methylation and increasing the expression of these two genes resulted in increased cisplatin sensitivity. Now, the European Association of Urology recommend neoadjuvant chemotherapy for muscle-invasive bladder cancer, especially the cisplatin-based combination. However, there is a problem of chemo-resistive patients, and there is no reliable biomarker for predicting the response of chemotherapy patients. The current approaches have several limitations. First, they don't guarantee 100% specificity. You can see that the recommended mutations can be found in the two groups, the chemo-resistant and chemo-sensitive, 'sensitive' patients. Also, the current approaches apply their experiments on a small number of patients or in vitro investigations. More representative samples is needed to generalize their results. Also, they didn't answer what is the best biomarker to identify the eligible patients. Are the genetic biomarkers sufficient, or I have to investigate for other biomarkers such as imaging biomarkers? Also, they should also take into account the suitability of a certain compound to the patient. For example, patient with impaired renal function cannot tolerate cisplatin. This factor should be also taken into consideration. Finally, the current approaches should be converted into clinical tools for doctors to use them. All the studies are just research that they convert into clinical tools. Thank you. Questions?
What's hypermethylation? Hypermethylation.
Do you know the methylation process?
Methylation. Yeah.
The incorporation of methyl group to a specific gene that might decrease its expression. Hypermethylation is increasing the methylation. It's the decrease in the expression level of a gene. If a gene has a hypermethylation, it will have low expression.
Okay.
So after this survey, have you set your side of your own research, what you're going to do, change this whole landscape?
Until I just made a survey about the current
program.
program.
I know, you did the survey. What do you plan to do after you do this survey?
Okay, we have now samples from, we collect samples to have our biomarker more reliable than the current approaches. But my work here is just to find the problem and start to solve it in the future.
Okay, I'm going to hand to Dr. Ruth for our next part of the session.
Okay, we've got 2 more talks left, and the next talk is gold nanoparticle conjugate cancer targeting aptamer and gadolinium chelate for MR cancer imaging. And the presenting author is UofL. Do you have your talk on there? I think done a good job keeping everybody on time.
Yeah? Let's keep that way. Yeah.
Good afternoon, everyone. So my topic is gold nanoparticles conjugate cancer targeting aptamer and gadolinium chelate for MRI cancer imaging. My name is Najwa Elbaz. I am a PhD candidate in the Department of Pharmacology and Toxicology, and I am a doctor So first, I have a background about the role of magnetic resonance imaging in oncology, as MRI has been used in diagnosis, staging, monitoring, therapeutic response, and treatment plan for cancer patients. It provides greater contrast within soft tissue in comparison to CT, and also it may reveal metastasis. So in most cases, before MRI scan, contrast agent is injected for improving visualization of tissue and organs, and what it does, it shortens the relaxation time of water protein within body tissue. And each MRI contrast agent has its own relaxivity, which describes its ability to changing water protein relaxation time and it correlates with its sensitivity. There are 11 FDA approved MRI contrast agents, and most of them are gadolinium based. And there is some limitation for the current contrast agent, as they can't be used in patients with compromised kidney function. They are not cancer specific, they have low sensitivity and specificity. So in our project, we developed a nano contrast agent. It composed of gold nanoparticles conjugate with gadolinium chelate, which is gadolinium DOTA, and cancer targeting aptamer ES1411. The core of the complex is about 4 nanometer, and each particle has about 10 ES1411 ligand and about 28 gadolinium chelate. And the total diameter of the complex is about 13.5 with negative 20.5 zeta potential. So ES1411, as Rajat mentioned before, it is a small DNA. It's G-rich 26 oligodeoxynucleotide. It forms stable quadruplex structure, and it makes it very resistant for cellular nucleases. It targets a specific protein called nuclein, which is expressed in cancer cells. And ES1411 has a thiol group to make it attached to the GNP. So in normal cell, nuclein is expressed inside the nucleus, and ES1411 doesn't cross the nuclear barrier. But in cancer cell, nuclein expressed on cell surface and in the cytoplasm, and it mediates uptake and trafficking of some oncogenic factors. When ES1411 binds to nuclein, it inhibits its function and correlates the anti-proliferative effect. So we use ES1411 for targeting cancer cell and also for killing cancer cell. And it shows the cancer selectivity because its uptake depends only on the surface nucleus, which is present only in cancer cell. Conjugation of ES1411 with gold has many advantages, as gold increase uptake by cancer cell and increase the potency against cancer cell up to 50-fold with maintaining cancer selectivity. Also, gold nanoparticle co-localize ES1411 with the gadolinium chelate. For MRI imaging experiment, we use two cell lines, triple negative breast cancer cell, MDA MED231, and normal mammary epithelial cell, MECF38. We treated the cells with three complexes composed of gold nanoparticles, gadolinium, and or ES1411, CTR, and PRO. CTR and PRO are they are small DNA. They are negative control for ES1411 because they don't target cancer cells. We use the 3 different concentrations of gold nanoparticles, and we use non-treated cells as control. After scanning with MRI, relativity enhancement of the contrast agent were calculated from this equation. For the result of the experiment shows that particles with ES1411, which represent the blue bars, have the highest relativity enhancement in cancer cell, and cancer cell in the left side of each figure, and normal cell in the right side of each figure. And the highest increase in relativity enhancement was achieved at 72 hours of treatment. So the conclusion of this experiment is that the proposed contrast agent with ES1411 shows selective targeting of cancer cells through accumulation and retention upon binding to nuclein protein. And the selective targeting has been validated by MRI scan. After that, we went to in vivo. This experiment was done using healthy Sprague Dewey rats, we divided the rats into 5 groups. 3 groups received the 3 complexes of GNP that I mentioned before. One group received multi-HANS, which is a current MRI contrast agent, and one group received saline, which is a negative control. The graph shows that liver and spleen has the highest concentration of gold in comparison to the other organs. And this is another experiment using tumor-bearing bulb-C mice. We injected 2 groups of mice with a contrast agent, but in this one we use different administration route, tailbone and retroorbital venous sinus. One group was injected with deuterin, which is a current MRI contrast agent, and the control group was non-treated. After 7 or 10 days, mice were sacrificed. We collected the tumor and organs, and the RCBMS analysis showed that liver has the highest concentration of particles. Spleen have some gold, but not high like liver. The tumor has a very little bit concentration of the gold. This means that the particle is being uptake by liver, which may affect its the targeting of tumor. So for this, we decide to address the impact of protein corona formation on GNBs. The protein corona is a protein that absorbed on the particle after administration. So this study showed that the particle with the AS protein 11 were incubated with human serum for a certain period of time. After that, the particles with protein corona were analyzed to find the kind of protein that is being absorbed on the particle. There was about more than 50 types of protein, but the most abundant protein are these 5. And one of them was the complement C3b, which is known as an opsinin protein. And opsinin means any antibody or other substance that bind to the foreign entities in blood and make them susceptible to immune system. So any pathogen is conjugated or absorbed C3b protein will be recognized with the complement receptor, which is expressed on the surface of macrophage cell. This is another study shows the uptake of particles with protein corona by macrophage cells. They use raw 264.7 mouse macrophage cell. They treated the cell with GNB AS protein 11 with and without protein corona, and also GNB Pro, the same with and without protein corona. The graph shows that the cell that is that been treated with particles with protein corona has the highest percentage of gold in comparison to the other cell that treated with particles without protein corona. So the reason for that is the protein corona has C3b protein, which is recognized by macrophage cell, and we know that liver and spleen are called rich organs in the macrophage cell. So the reason for this is the reason for uptake the particle by liver and decrease the particle concentration in blood before reaching the tumor site. So in order to decrease the amount of opsinin protein, we add polyethylene glycol to the particle, which is called the degylation of particles. And polyethylene glycol provide hydrophilic protective layer around the gold nanoparticles, which decrease absorption of opsinin protein, which is the first step in the opsonization. Also, PAG increase the solubility of particles in buffer and the serum, and decrease particle aggregation. Also, PAG has a thiol group that binds to gold nanoparticles. So my future work is develop different size of gold nanoparticles and conjugate them with DOTA, PAG, and or ES1411 and Pro. I will determine the absorption of opsonal protein on the particle by measuring the hydrodynamic diameter of particles after incubation with proteins, blood proteins. Determine the uptake of incubated particles with blood protein by macrophage cell and some malignant cells. Also determine the uptake of particles by tumor cells. So my acknowledgement to Dr. O'Toole, Dr. Bates, Dr. Malik, Dr. Kenton. My acknowledgement to Rajat because he did all the particle synthesis and the characterization. Curtis, he was helping in animal study. Daniel, he helped in cell study. I would like to thank Excite Grant for funding and IPEPS for supporting me. Thank you.
Open for questions. Nobody? Okay, thank you. So this is the last talk of the session. The title of the talk is Lung Cancer Survival Prediction via Machine Learning Regression Classification and Statistical Techniques. Dr. Herman Freibus is going to present this, and Dr. Freibus from the University of Louisville.
Thank you for coming to this last talk in this session. And so what we were trying to look at here, and we're still trying to understand better, is can cancer survival be better predicted than what is currently done by clinicians? So it is notorious how the survival of patients with cancer is a highly inaccurate science. And in particular here, focusing on lung cancer, which is the most number of cancer cases, shows that in general, physicians and clinicians tend to overestimate short survival of patients and underestimate the long survival. And for example, here for patients predicted to live longer than 3 months, only about 60% of them actually survive that long. And this, under another example here from the literature, when physicians predicted survival to the month, only 10% of the time. And within 3 months, about 60% of the time, and 4 months, about 71% of the time. And so why would this be a problem? Because it helps to actually plan treatment better, and also for people who have this disease to plan their lives better, if this could be done more accurately. So the idea is here to apply some sort of learning algorithm to help doctors and even patients to figure this out a little better, so to complement this knowledge that they have. So the idea is that with machine learning, the algorithms can be used to do this more efficiently and more quickly, and ultimately more accurately. So lung cancer is the leading cause of cancer death, and a few models in the literature actually focus on lung cancer in terms of trying to predict outcomes. And currently, most survival models are classification based. So some of them predict survival or non-survival. Some of these models have varying degrees of accuracy. It's a wide range. in some cases, regression is more appropriate than classification for survival time, since time is a continuous variable. Previous work has used the SEER database to actually analyze large data sets of patients. So this database is open to all researchers to look at data of cancer patients going back almost 50 years, or even longer actually in some cases. And so previously we had developed a regression-based model which used 5 different types of machine learning algorithms plus a custom ensemble to try to predict survival time. And there were 24 patient parameters that were known at the time of the diagnosis. And these models predicted within a relatively narrow range because the data was kind of unbalanced, and also had large root mean square errors, or RMSEs. Also, we applied an unsupervised, actually not just one, but several unsupervised methods to see if the survival could be predicted in this way. And also not very successfully. So if you expect this presentation to break this problem, it's not true. It's still not going to be very successful either. But it's worth trying different approaches and see what does it take. How does the data have to be arranged? How do the parameters need to be selected to make this more accurate? So in the current study, first we used a statistical analysis to select significant parameters from the patients, and also to reduce the dimensionality and the noise with the model. And we used a combination of classification and regression. First, a classification to separate the data into 3 categories of survival time, like a short-term, medium, and longer term, and then use regression modeling to predict within those categories how long these patients would live. So the data itself, as you can see, is difficult. We were able to select about a little over 10,000 records, and the mean was about 20 months of survival. The standard deviation was almost 17 months for that survival. So this is crazy, right? So we applied an ANOVA analysis, and with these 24 input parameters, and to try to figure out these 4 intervals. And we removed some outlier data, designate the parameters as either numeric or non-numeric, and then used some statistical programming here with Minitab to improve the linearity of the probabilities. So out of the 24 parameters that were selected, 15 were determined to be significant through the statistical analysis. And the parameters were not selected if they could not be estimated, or they could not be known at the time of diagnosis. So they were after the fact, because the idea is to predict things, right? So out of the 15 significant parameters, 13 were selected for the use by the model. And I'm not going to go through all of these parameters. We don't have time, but it's detailed in the paper. But as an example, for example, there's data about the location of the primary site, the number of primary tumors. There's data regarding surgical procedures that the patients may have had, and the tumor grade, the tumor stage. So these were the ones that were selected to be significant by the statistical analysis. So the classification exercise created these 3 groups of patients. And we used the random sampling to select 75% of the data for training, and the remainder was used for validation. So the validation data was used by each regression model then within each category. For classification, we decided to use random forest as an algorithm. And for regression, we picked these 3, again, random forest, gradient boosted machines, or GBM, and general linear regression. So we trained it a little bit outside the category because the classification is not perfect. So kind of slopping over on both edges. And so it's able to predict within or slightly outside the category. So one group was less than or equal to 6 months, the training data for this was 0 to 12-month survival. And then there's a group for 7 to 24 months, which use all of the training data. And then greater than 24 months, with the training data being greater or equal to 18 months of survival. So with the random forest algorithm, which was used for classification first, and then later on as one of the regression algorithms as well, essentially it creates these decision trees that recursively split the training data, and it could be described as a simple heuristic ensemble of decision trees. And these were the parameters that were chosen for it. And for the regression, these were the parameters here. So I'm not going to describe them in one by one here. Okay, and then for the gradient boosted machines, which was used for regression only, it's similar to random forest. It creates an ensemble of decision trees, but the model is built progressively instead of all at once. And it actually, in theory, is supposed to learn from the previous trees when building new trees in order to minimize the error in the subsequent trees. And these were the parameters for this algorithm. And then a general linear regression, which doesn't have unique parameters. And we also applied a custom ensemble combining these three, weighing them with different weights. And so the weights depended on how well each one of these models could predict the survival. So for the classification, which was done with random forests, this was essentially what's called the confusion matrix. And the overall accuracy was about 50%, and an error rate of, which is the other half, 50%. The sensitivity differed widely depending on which one of these classes we're talking about. Class 1 being the short-term survival, class 2 being the medium-term, class 3 being the case of greater than 24 months. And these measures, these kinds of analyses, may not be the best to evaluate classification models, especially with a set of unbalanced data as this one. And also, it was noted that the classification model is overfitted in class 2, which is the 7 to 24 month survival, when it used the bulk of the training data. So for the class 1, which was the short-term survival, the best performing model for the regression now was the random forest. And but all of the models actually predicted within the range of 3 to 7 months, and under-predicted longer survival times. For the second class, which was the 7 to 24 month survival, the best performing case here was the GBM. But although it had the best root mean square error, the random forest actually performed better, as can be seen with these comparison plots. And this actually points out some of the limitations of using any one measure to figure out which one of these is better than the other one, especially when the data is this complicated. So for the third class, which is greater than 24 months, the random forest performed better, but in terms of RMSE, but in the comparison plots, the GBM seems to be performing better. In general, however, the regression models seem to be more accurate for the lower survival times, and as can be seen, the progression of these root mean square errors in the comparison plots. And so this root mean square error, this RMSE, was used because it is a common measurement of accuracy, but it can be misleading, especially because larger errors can have larger effects. It can also be inflated when predicting smaller times, and by itself, it does not reflect the performance. Also, the classification needs improvement, as you saw from the matrix. If it is not properly separating these patients into these groups, then it leads to less accurate regression. Potentially, it could be different sets of classes that would make it more accurate. But to do all this, it would be ideal to have a more balanced data distribution. to avoid overfeeding. So in comparison to the previous study that we had published, this was actually an improvement, if you can believe that. Because in the previous study, gradient boosted machines achieved the highest root means per error was 15 months. In this case, in the current study, it was random forest, it was 10 months. So primary limitations include that the range of the data set is too wide. Not all the patient deaths that are recorded in the database are necessarily due to cancer. They might be dying from other reasons. And longer survival time means that other causes of death are more likely. So in conclusion, we can say that regression models have potential to accurately predict cancer survival, but mainly for shorter times. In that case, they're not necessarily better than the doctors. However, more work is necessary to improve and validate such methods. So we can't give up yet. I'd like to thank my students, Chip and Benas, as well of course my co-author, James Andrew, who worked on this, and then my collaborator, Dr. Van Berkel at the UofL Medical Center, and also partial funding by the National Cancer Institute. Thank you.
How do you make a lung cancer data set more balanced? That's going to be a constant limitation no matter what you do, right? Because you can't change the incidences of cancer.
Right.
Are you referring to maybe increasing the different features that you look at, or is that not?
That would be one thing, or use a different data set. This is a particular data. The SEER database is in a particular group of data, but there are other data sets as well. So it would be interesting to evaluate how they compare to each other with these methods. There's also the possibility of potentially, with lung cancer, there's a specific data set that could be used which we have not tapped, which seems to be, at least at first glance, seem to be more balanced than this data set. Yes.
So I have two questions. The first one is regarding the feature selection. You just used ANOVA for selecting the most significant feature. Did you try any other feature elimination or feature selection algorithm? Or?
No, that's the one we tried
Yeah,
here.
because, okay,
But
so
we could try other ones, yes.
yes.
Yeah, the previous study, we actually didn't do that. It was based on what we felt was biologically relevant.
Yeah. The other one, when you do both classifications and regression, when calculating the error, you consider the classification error while calculating the regression error, or they are completely independent?
Here we did it independently.
Independent. So if it's misclassified in the first stage, still
That's
the
right.
regression could
That's
be
right.
valid and accurate.
Yes. Dr. Elbas, your question.
Yes. If you need to improve the accuracy of prediction, your opinion is this is need more feature, or you need more classification model like deep learning or random forest or OSCP more complicated, or you need more data sets? So what's your opinion like?
I think it's all of the above. Yeah, because it's not going to, I don't think it's one single thing that's going to make it a breakthrough. Yeah. Yeah.
And, good presentation, Herman. So have you considered doing a power analysis to determine what the sample size, sample distribution should be?
That would be a next step, actually. Yeah, we had kind of discussed that, but then my student graduated. But yes, it could be taken further. Good point.
Let me ask a different question. I try to poke some fun to entertain the audience.
Let's not start now.
At least not with you, right?
So the question is, let's just say, forget about these academic numbers, let's forget about the statistic for now, say, if every time, I don't know if you guys follow, if Wall Street people talking about picking stock with only 50% accuracy.
Yeah, that's my question.
And then people would say this is the same as, you just use a monkey. So I want you to comment on You know, you spend so much time and lost the students for graduation. You're not doing better than a monkey. So let's entertain that idea, entertain the audience. I want to hear your argument, how we can do better.
Then monkey.
50%, that's one of
No,
them.
it's 50.3, so there's a 0.3% improvement.
Well, I think it goes back to some of the others like Dr. Al-Abbas mentioned. Yes, we have to explore further.
No, I guess first level question is say, when we have 50%, I know, I mean, scientifically you say, yeah, we made a progress, but is that true value in there, so only have 50% of accuracy?
Not clinically, no.
That's right, that's my argument, I guess, yeah.
Also, one more thing regarding 50%, you have more than 2 classes, right? You have 3 classes for the classification, right?
Yes,
Not
three.
3 classes. So the baseline is 33, so 50% is above the random. The baseline is not 50%. It's not a binary classification, so it's above the random by about 17%, I think, right?
Yeah, it's 3.
3 classes, so the baseline for random guess is 33%.
So maybe it's a chimpanzee.
Much above the random selection.
But the sensitivity and specificity values are in the 30s.
So I didn't see the sensitivity and specificity,
so
but the
that's
accuracy is above the.
Yeah.
Most likely the sensitivity and specificity will be left and right from the accuracy,
Yep.
so it will be in the 30s and the 70s, 60s.
That's what it was.
Yeah,
Yes.
yeah, exactly. So it's some sort of above the random.
It's,
Not
yeah, it's a little
completely
bit less
random.
than right. It's more like a monkey who went to school.
That's kind of entertaining.
So anyone else have any comments on monkeys?
Let's thank Dr. Ferguson. That's the end of this session. I guess we're